very much. So since we are absolutely on time, we have time for an extra presentation, which is not properly a pitch on a special cold topic, but is more a presentation like an offer from a newcomer in the ETPN, which is oncome. So Maria, if you can come. They became ETPN member, this week. So thank you for that. So oncome is a group from the, let's say, Hospital University of Santiago de Compostela.
Yeah. So thank you very much for the opportunity. We have, as he said, become members this week. And I would like just to introduce ourselves, so maybe you can consider us for some potential collaborations. We are based at the University Hospital of Santiago, and we are a mixed group. We are clinicians and also researchers. And our main focus is the treatment of metastatic disease. So we want to develop personalized medicines and try to find new options for metastatic cancer patients. So the group is, since clinicals are involved, we are really translational. We aim to provide real solutions, and we are structured in 2 basic research labs and 2 transversal platforms, liquid biopsy and also epigenomics. And we also have some clinical trials undergoing. So I just going to introduce you very briefly the labs. So on the one side, we have the cancer modeling lab. This is aimed to, the main activity is to try to understand why metastases occur and try to identify novel therapeutic targets to provide solutions. So we work in this line to develop more sophisticated in vitro cell culture models in 2D and also in 3D, and also trying to find novel models of interest, and we are especially dedicated to zebrafish models because they are simple, more versatile, and they are an alternative and complementary to traditional mice models. Then we have the nano-oncology group, which is the one that I'm leading. And we also develop nanostructures for personalized medicine. We try to find, making use of the opportunities that we have with clinical samples and so on. We try to identify novel biomarkers that allow us to target specifically our nanoparticles to metastatic cancer cells. And we have different platforms, biocompatible, and we can decorate them with different ligands to target them to these cells. We have already identified 3 interesting novel receptors that we are trying to exploit and see if we can provide a better targeting. We also work in gene delivery and siRNA, microRNA, long non-coding RNA delivery, because we think that personalized medicines should also consider all these genomics and so on. And we have a more incipient research line for nanotheranostics. So we are collaborating with Fernando, who presented the work yesterday for PET imaging, and we are also interested in other modalities of imaging. And we have now some multimodal nanoparticles that we think that have a lot of potential for diagnosis, improving diagnosis of metastatic disease, and for personalized medicines to try to see if the therapies work or not. Then there is the liquid biopsy analysis unit, and is dedicated to explore biomarkers that are present in fluid bodies like plasma and other bodies like circulating tumor cells, which are related to cancer prognosis and response to treatments, and also ctDNA, microRNAs, and exosomes, and so on. So this is more transversal and is giving support to the clinical trials, but we can also have a lot of information about it, and there are also nice collaborations, for example, as Lorena presented with the INL, to develop novel methodologies for isolation of these biomarkers that overcome some of the limitations of the currently available techniques. There is this epigenomics unit, which is the newest group in the lab in the group, and is dedicated to the epigenomic analysis, also related to metastases and this. So all together, we are trying to cover the different parts, from discovery of novel biomarkers to understanding which are the therapeutic targets and developing nanomedicines that can help to to improve therapy and diagnosis. And because we are a clinical group, there are also a clinical trials unit, and this is really clinical, and there are different clinical trials, mainly in breast, colorectal, and lung cancers, but also some in pancreatic cancers, and so on. So this was just because we are new members. I would like that you know us and consider us for some potential collaborations if you are interested. I forgot to put my email address in the presentation, which is a big mistake, but I will be around the whole day, so if you want, I can give you the details. So thank you very much for your attention.
Well, Maria, thank you, and welcome on board. That's cool. I will just finish with a quick announcement, and then we'll have the coffee break. The announcement is quite simple, is that the new call for application of the EuroNanomed 3, let's say, ERANET, will be open in a few days. You have to be really, let's say, reactive, because the call are going to be announced in November, and it's going to be really open for pre-application in January, and the final deadline for final submission will be in May. So basically, there are many more, excuse me? For pre-proposal, yes, and then the real one is in May, right? So just to let you know, the best thing that you can go on the website of EuroNanomed, and just to show you the list of eligible countries. I think there are 19 of them, which is quite a record. So you should go on the EuroNanomed website and check your national content points. The new call is covering the whole things that you do, because it's about regenerative medicine, targeted delivery system, and also medical devices. So it's quite broad. You have many countries involved. We all know that the rate of success is quite higher than in H2020 call, so you should definitely go. This is clearly a nice opportunity for funding for your collaborative project, okay? So I recommend you that you go on the website, and I recommend that you have a coffee right now. 30 minutes. We are back in 30 minutes with the presentation of the Nano 4P flagship